Hello friends, now I would like to discuss with you microbiology, mainly viruses. In the viruses, I would like to start today with rabies virus. Rabies viruses belong to the family rhabdoviruses. So in the rhabdoviruses, we would first discuss about rabies. I would just like to give a brief introduction of all the viruses and bacteria in microbiology, and we will deal in detail all the pathogenic features and etc. in general medicine. So, rabies virus. Rabies. If anyone thinks of the word rabies, especially I think of dogs, because it's most commonly transmitted by dog bite. So how does the rabies virus look like? So for this, in viruses you will learn about morphology. Here the rabies virus is a bullet shaped virus. It will be somewhat like bullet shaped, perfect bullet, and it is bullet shaped, and it has with one end is rounded and the other end is planar or sometimes concave. It has an RNA genome which is single stranded and negative RNA genome. So this is RNA genome, and this RNA genome is covered by a matrix protein layer, right? And above this matrix protein layer there is a lipoprotein envelope. This is lipoprotein envelope, and to this lipoprotein envelope there are peplomers which are attached. And in the matrix there is an RNA dependent RNA polymerase. 1 minute, yeah. There is an RNA dependent RNA polymerase in the matrix. Fine. First let us label it. So this is RNA genome, RNA genome, right? And this is RNA dependent RNA polymerase. Covering this genetic material there is a matrix protein layer. Beneath the matrix protein layer there is a lipid envelope, or to say lipoprotein. It's not just lipid but lipoprotein. An envelope, and to this lipoprotein envelope there are many spikes which are peplomers. This is the morphology of rabies virus, right? So one end is bullet shaped. This is mainly bullet shaped. One end is, you can see this end is convex in shape or round, and the other end is planar or sometimes concave, right? So let's discuss about each and every part in detail. This RNA genome, it is single stranded RNA, that is negative sense strand, and it has helical symmetry. You can see it is like a helix, right? And this is covered by an envelope which is matrix protein layer. Beneath this matrix protein layer there is lipoprotein envelope, and on the lipoprotein envelope there are many peplomers. This is the morphology of rabies virus, which is really very simple. And then resistance. this rabies virus is resistant. Here you will be dealing with what is resistant, what is sensitive. It is resistant against dryness, cold, decay, and it is sensitive to phenol, formalin, beta propiolactone, sunlight, UV light. So what is resistant? Resistance is it can't be killed by dryness or cold or delay, but it can be killed easily by phenol, formalin, beta propiolactone, sunlight and UV light. The main important things are antigenic sensitivity properties. Each and every virus has his or her antigenic properties. These antigenic properties include: first, surface spikes. These surface spikes, these are the spikes, right? These surface spikes have glycoprotein. Glycoprotein is with the protein along with the carbohydrate residue. This glycoprotein is strongly antigenic, and antibody is proteins. So this is used in vaccines. And antibodies are produced, these are used in vaccines. So mainly one of the properties is surface spikes. These surface spikes have glycoprotein G, which is strongly antigenic, and this is used in vaccines. The second one is nucleoprotein. Nucleoprotein. This nucleoprotein also induces antibodies. These antibodies are not protective, so these are not used in vaccines. This is the second antigenic property which is present. And the third is hemagglutinin, which is one of the peplomer. Hemagglutinin, which is a peplomer. What does it do is it easily aggregates the RBCs. And the others are membrane proteins, glycolipids, RNA dependent RNA polymerase. All these are antigenic properties of rabies virus. So first mainly morphology we have learnt, and then resistance, and then antigenic properties. Antigenic properties mainly include surface spikes and nucleoprotein, then hemagglutinin, and then membrane proteins, glycoproteins, RNA dependent RNA polymerase, and etc. So what is the pathogenesis of rabies virus? Here I just wanted to give an outline of each and every virus so that it becomes easier when we learn everything in detail in general medicine in our future classes. So first, pathogenesis. So this pathogenesis, I just give a flowchart for this. I usually prefer flowcharts for all the pathogenesis. I'm sorry for the inconvenience again. So this is for this pathogenesis, there is rabid dog. Whenever this rabid dog is infected by rabies virus, the dog which is infected by rabies virus is a rabid dog. This rabid dog bites humans, and this rabies virus enters wounds Whenever the dog bite, it produces a wound, and through that wound rabies virus enters, and then it multiplies in muscles and connective tissue, and from the muscles and connective tissue it spreads to central nervous system, and from there it again spreads to peripheral nerves, skin and adrenal glands, and thus manifesting its main symptoms. All these are the pathogenesis of rabies virus. Rabies virus is mainly a neurotoxic virus to say. This rhabdovirus, dog which is infected by rabies virus, this bites humans. Whenever it bites humans, the rabies virus, through the wound it enters muscles, and then through connective tissue, and then spreads to CNS, and then from there it spreads to peripheral nerves, and then skin and adrenal glands. So this is the pathogenesis of rabies virus. So where will we culture this rabies virus? So in olden days, as viruses evolved, viruses cannot grow in any of the cultured medium. So we started them growing in animals. So first let us learn the animal susceptibility of rabies virus. So animals, this rabies virus mostly grows in warm-blooded animals. Mainly warm-blooded animals, like mainly man, dogs. These both are mainly intermediate hosts for rabies virus. If we inject or infect an animal, any warm-blooded animal, most probably on what will we do experiments? It's probably mice. If we inject or infect any animal, especially mice, then this leads to encephalitis, most probably. And this encephalitis is also caused by intracerebral inoculation. If you directly inoculate it in the intracerebral cavity, even then it causes encephalitis. So here it occurs in animals, and it is mostly occurs in warm-blooded animals. And what is this is what through this we can grow in the animal bodies and then find it. And there are other ways of culture too. And in the animals, if this rabies virus can be isolated from animal or human infection. This virus is called as street virus. If it is isolated from animal or human infection, then it is called as street virus, and this causes fatal encephalitis. So encephalitis is nothing but brain parenchymatous infection. So in the brain we find intracytoplasmic inclusion bodies, which are called as Negri's bodies. These are seen. In the brain parenchyma, we see intracytoplasmic inclusion bodies or Negri's bodies. So this is the main diagnostic feature of rabies virus, presence of intracytoplasmic inclusion bodies or Negri's bodies. And thirdly, the changes, some of the changes are seen. Some changes are seen by serial intracerebral passages in rabbit. That is, if you serially inoculate it in the rabbits, then slowly it goes through the brain parenchyma and it causes some changes. So this virus is called as fixed virus. Whenever the rabies virus does this thing like slow in changes, then this is called as fixed virus. So here during this time the virus will be more neurotropic, and thus it again leads to fatal encephalitis. And Negri's bodies, which are characteristic features, are not demonstrated in this rabbit. I think you got it. It's like all animal susceptibility is mainly of three types. One is in warm-blooded animals like man, dogs, and warm-blooded animals. Mans and men or dog are intermediate hosts, but mostly we inject or infect the rabies in animals, that is, which causes encephalitis. And this rabies virus is isolated from animal or human infections, so it is called a street virus. It causes fatal encephalitis. Encephalitis is brain parenchyma infection. When you take the cross section of the brain, then you can find intracytoplasmic inclusion bodies, which are Negri's bodies. And if it is inoculated into rabbit, then some changes will be seen, because in the rabbits it slowly goes through the intracerebral passages. So this is called as fixed virus. Here it causes fatal encephalitis, but it does not produce any Negri's bodies. All the reasons for why not producing and everything will be dealt in medicine class. So, which is tomorrow, most probably. Next, inoculation. That is mainly cultures. One is animal susceptibility and the other is culture. Culture is, there are mainly few modes of culture. One is cheek embryo. In cheek embryo, inoculated in yolk sac, live attenuated vaccine strains are developed through this cheek embryo, are developed by this method. So one is cheek embryo, that is in yolk sac, and the other is tissue culture. In tissue culture, the different specimens which you use is cheek embryo, fibroblast, hamster kidney cells, human diploid cells, Vero cell culture. All these are the cultured mediums which are used. In the next class, we will learn about the lab diagnosis of rabies virus in an elaborate manner, and then we will learn about vaccines and complete elaborate study of rhabdovirus. This is just an introduction class of rhabdovirus, where we dealt with morphology of rhabdovirus, then resistance of rhabdovirus, antigenic properties of rhabdovirus, pathogenesis of rhabdovirus, and finally animal susceptibility. I know this is little confusing and you're not clear with it, but I'll clear it in the next two classes. And cultures which are used in the rhabdovirus. So please don't forget to comment it. Okay, bye.